Hey guys, this video is the beginning of chapter 15. We'll be going over the first couple sections of chapter 15 on genes and proteins. So let's talk about central dogma just one more time, that we are going to have DNA and it is going to be transcribed into RNA, and then that stuff is going to be used and translated into proteins, amino acids that will make proteins. So that is central dogma right here. So central dogma, we have the DNA. It's going to go into RNA. It may be processed, it may be not. If you're a eukaryote, we're going to talk about processing. If you're a prokaryote, transcription and translation is happening all in the same place in the cytoplasm and almost at the exact same time, which is pretty cool and very efficient for prokaryotes. Eukaryotes were complicated and it's not exactly the same process. Basics the same, but some details are different. We're going to go into that. So transcription is writing your message. This is transcription, writing your message, and then you created your messenger RNA. You're going to use that messenger RNA, and now you can translate it into your protein. There's just one amino acid, but it's going to amount up to lots of amino acids that make the protein. So central dogma, DNA, RNA, amino acids for proteins. Speaking of the amino acids, I want to say we covered this before in our macromolecules chapter, but here they are one more time, where they are all going to have the same head. They're all going to have this amine group and an acid group, so they are amino acids. The tails differ. The tails are going to differ, so they are classified on their different tails. Are they nonpolar? Are they polar? They have a positive charge. Do they have a negative charged tail? And then later, if you ever do advanced biology, you can go into proteomics. We can talk about phage analysis and all that good stuff, but we don't talk about it here. We're just going to talk about the basics going on with central dogma and how we get from DNA to RNA, and then what do we do with that RNA. That's what we're going to talk about. So all of those different amino acids are going to be produced. We only have 20 here. So all the proteins in your whole body are made out of these 20 amino acids. And how do we code for those? We are going to code for those using the DNA that we have. So DNA is going to have 5 nitrogenous bases, right? There's adenines and thymines, and there's cytosines and guanines. We have A's and T's, we have C's and G's, and if we are making RNA, then you would swap out the T with a U for uracil. So we are going to take 3 of those nucleotides. So we're going to take our double-stranded DNA with these guys buddied up like this, like we've seen in all the pictures. And we're going to take 3 of those nucleotides. So we're going to unzip our DNA. We're going to have to unwind it. And we are going to, based on that single strand of DNA, that side of DNA that we're going to call our template strand or our nonsense strand, we are going to match up. And it just matters on what nitrogenous bases that we have. We have A's, we have T's, we have C's, we have G's. Whatever we have, we're going to pick 3 of those. 3 of those are going to be called a codon. So if you do the math, and we've got these 4 bases, and you can do the math and come up with that, we can get 64 different combinations if we have 3 spots to place those letters. Maybe it's CTC, maybe it's AUG. It could be any of those. So there's 64 total combinations that you can make, and there's only 20 amino acids that they produce. So since we can create more combinations for one amino acid, like if you look here, here's lutein, and you can see there's 4 different things in this box here. That's called degenerate. It's degenerate. So there's a little bit of wiggle room, basically, when we're coding for these amino acids. So how do we code for these things? You're going to use this box, and it's exactly what you're going to do for the plasmid project. which I have updated, by the way, on the instructions. So please start looking at that. It's due in 2 weeks. Start looking at that and let me know. This is question number 7. So this is how this would work for question number 7. Hint, hint, wink, wink, nudge, nudge. How do you use this codon table? So say that we have a codon and it has the letters, oh, I don't know. What do we want to say? Let's say, what are we going to make? We're going to make, well, we don't have T's. Let's say that one, right? Yeah. We'll swap out our T's with our U's because RNA is not going to have U's. So if we had a C, a U, and a C, how does that work? So we're going to look for the first letter. Here's a C. And then the second letter is up here. So it's a U. And then our third letter is a C again. So third letter in this column here is a C. So here we have C, U, C, and it codes for this amino acid. It codes for lutein. So that's how this codon box works. You are going to have your 4 columns. This one's DNA, DNA. This will be non-template, template. Then you're going to have your RNA, and then your amino acid codon. What are you coding for? So you will write, beginning at your gene for insulin. Sorry, I'm blanking. Based on your insulin gene, you are going to go with, first letter, second letter, third letter on the non-template side of it. Then you're going to go with the matching first letter, second letter, third layer of the DNA on the right-hand side of the DNA. So this will be left-hand side of the DNA. This is right-hand side of your DNA. That's going to be our template that we're going to use. That would then make RNA. And remember, if it's A, it's going to code with a T, but instead of T, because we're using RNA, it would be U. If you had a C, then you would put a G there. If you had a G, you would put a C there. If there was a T, you would put an A there. So then you would make the complementary RNA strand here in these things. And based on those, then you are going to code for whatever that is. Just a hint, there are significant codons that you need to know about. So the first one is start codon. This is the first one that starts off all the other ones. So your first codon should be thymine. It should be AUG. So I want to say that's given for you in the table for question number 7, and you should code for, it should be that guy in that first box that you are coding for. Hopefully this is making sense. Please watch the video too on how to construct your plasmid. That is in the D2L thing, but we are going to start every protein is going to start with this start codon, every single one. So there is one start codon, he's right here. He starts off every protein going on. So we're going to code for this guy, and then we will go in units of 3, this codon, then this codon, and this codon, and this codon. And eventually, when we finally get to the right coding, it's going to code for a stop codon. So the stop codons, there are 3 of them. They are also called nonsense. So the 3 nonsense or the stop codons are this one, and this one. UAA, UAG, and UGA. If you hit those are immediately halting transcription. You are done writing your message, and then we are going to finish. So again, just to summarize, there are more codings than there are amino acids. So different amino acids can have up to, 4. There's 2 more for lutein. So we've got at least 6 different combos that'll code for that one amino acid right there. So it's called degenerate because there's more combinations than amino acids. There is one particular start codon that will start off this whole process. You will code for however many amino acids you need in that gene. As you get down to however many it is specifically for insulin, you will code down to how many there are, maybe 50 something. But you'll get down to the end, and at the end of your table, if you don't see a stop codon at the bottom of question number 7's table, something's wrong. And you need to go back and see if maybe you've skipped a letter when you are chunking out your codons. If it helps, take your strip, take your strip of paper. I'm eating sushi, so I got my sushi chopsticks. Take your strip of paper and mark it every 3, and just mark them off every codon that you've got, and that should help you fill out that table when you are doing these. The start one should be, you should have AUG as your very first codon, and then go all the way down the gene of insulin. And when you finally get to the bottom of the last 3 boldface letters, those last 3 boldface letters should be a stop codon. And that's how you know that you have transcribed this correctly in your question number 7 table. It takes time, so please start on that now and get going. If you have questions, again, email me, drop by on office hours in Zoom. Let me know if you got questions. So this is how the codon table works, how you can read it. You will know it, love it in a short amount of time very soon for this project coming up. Another place that you can play around with are the animations. So blow this PowerPoint up on the slide view, click the link, and you can play around and see what codes for stuff. So maybe that'll help reinforce how to use the codon table to go from RNA to whatever amino acid you are coding for. We mentioned mutations before, and this is exactly what I was talking about. Based on those 3 letters, what is in that codon, you will code for an amino acid. And if you change up those letters, then you end up with mutations. They could be point mutations where just one nucleotide is different, and that could code for a different amino acid, like in missense, where we missed one, the letter was different, and it coded for a different amino acid. So instead of this guy, that one letter switch gave us a totally different amino acid. That's a missense mutation. Sometimes if we miss a letter, it's still okay. It's a mutation, but it doesn't show up. So we call that one silent. Other times we missed a letter and we accidentally coded for a stop when we didn't want to. We stopped too early. It's premature. So we had a nonsense mutation because this was supposed to be tyrosine, and instead we stopped it. So these are the 3 different point mutations where it's just one letter that's off. On a frame shift mutation, those are insertions or deletions. So they can shift the entire rest of the nucleotide sequence down or forward a bit. If you deleted it, you're going to shift them up. If you insert them, you'll push them back. But either way, every codon after that is messed up because of that frame shift mutation. So they involve insertions or deletions, and they will change everything after that. So that's why the whole thing is called frame shift. You've moved everything, and you code totally different amino acids because of it. So those are mutations. Let me know if you got questions about those. The textbook mentions a question, and it says kiwis are 6N and strawberries are 8N. Who has more DNA? And initially you're like, oh, 8N, it has to be the strawberries, duh. But it's not, of course, because they set you up for a trick. So we have kiwis, and I had to look it up because I'm like, well, it's 6N. But what's the N, right? The N's the set that you have. And for us, we're 2N, and our N is 23. 23 from mom and 23 from dad. And in this case, kiwis have 6 N's. What is in their N? Each N for them is 29. So if you have 6 times 29 chromosomes, 6 sets of 29, then you end up with 174 chromosomes. Let's check out strawberries. Strawberry is 8N, and that sounds like so much more. How much is an N for them? How much is a set of chromosomes? Their one set of chromosomes is only 7. It's only 7. So when we do the math, 8 times 7 is going to give us 56. So kiwi is our winner in the most DNA challenge, even though it looks initially like kiwi didn't have as much DNA. It has way more. Okay, let's do transcription for prokaryotes. Let's talk about that business. Transcription is going to be 3 steps similar to our DNA replication. There is going to be initiation, there is going to be elongation, and there's going to be termination. So when you initiate transcription, you are going to have to unwind that DNA bubble a little bit, unwind the DNA, and create a little bubble in there, your transcription bubble. So we're going to unwind it and break apart, bust those hydrogen bonds, and unzip the DNA in that little section called a transcription bubble. And then you're going to use that DNA to attach on your RNA. So it's going to attach on to a place called the promoter. So that is a chunk of DNA that are specifically coding for RNA polymerase to attach on is called a promoter. Either it's a prokaryote or eukaryote, but they're all going to have this area right here in this -10 region called a Tata box. Both prokaryotes and eukaryotes have this gene coding. So that's part of the reason why the RNA polymerase, which lays down new RNA, DNA polymerase lays down new DNA, right, on DNA replication. RNA polymerase is going to lay down new RNA. So with RNA polymerase on prokaryotes, there's just one of them. And it knows where to bind on because there's a Tata box there. It says it is -10, and over here it is +1. So we're going to call all of these guys, these are downstream, and this is where we're going to have the genes that actually get coded into RNA. And this is going to be upstream on this end of it. So we have upstream and we have downstream. Upstream have these negatives. So from this point here, we're -10 this way, and now we're -35 on this way. And these special coding regions are called the promoter, where the RNA polymerase is going to bind and begin at the initiation site, where we can now start to lay down new DNA. There are different subunits of RNA polymerase. There's 5 total. There's 2 alphas and 2 betas, and there is a sigma. Once the sigma signs on, that is what is going to begin the elongation and begin laying down of new RNA and start the next step of elongation. So RNA pol has bound in initiation. It has all the subunits. It bound at the promoter region that has the Tata box in it. It tells it where to bind. It's going to latch on, and once everything is ready and good to go, it's going to start sliding down the DNA like a choo-choo, and it lays down RNA, just like we said DNA pol lays down DNA. RNA pol lays down RNA 5' to 3', 5 to 3 every time. So it's going to come shooting down and laying down this new messenger RNA 5 to 3, and continue on until it gets to a termination signal. That termination signal at the very end, it is going to be based, we have termination based on a DNA sequence, or it could be a row protein here that will stop it. But either one of those will cause termination, row dependent with the row protein, or it could be row independent. That's going to cause the RNA to do a hairpin turn, and then it terminates at that point. But that termination is step number 3. It's the end of creating the message for prokaryotes. And remember, prokaryotes, where is this happening? When we compare prokaryotes to eukaryotes, I don't think I've done that yet. But where prokaryotes' DNA is housed, remember, it's not a nucleus in a prokaryote. They don't have that membrane-bound nucleus. It's in that area in the cytoplasm called the nucleoid, just hanging out there. So because it is open access, they can transcribe and translate much faster and easier than we can. So literally, here is your DNA in this green strand. All of this is DNA. And as it is being transcribed into messages, we're going to transcribe, and we're creating, every one of these blue lines is messenger RNA. We can also start translating those messages into proteins. So we can have amino acid proteins coming off, proteins coming off, proteins coming off. every single one of these ribosomes are going to attach on, that's all these little ribosomes, and they will start creating proteins coming off. So transcription and translation are both happening in the cytoplasm. Transcription happens first, of course, but it gets translated really fast. Some of this RNA, its shelf life is not very long, like seconds. So this is happening really quick in the prokaryotes. Eukaryotes, transcription on eukaryotes, basics are still the same. It is still initiation, it is still elongation, and it is still termination. Again, location is one of the differences. Our transcription, the writing of the message, happens where the DNA is, and for us, it's inside that membrane, inside the nucleus. So we have to transcribe the message inside the nucleus, get that message out through a nuclear pore and into the cytoplasm to meet up with the ribosome. So our RNA has to, our messenger RNA has to live longer than just 2 seconds. It has to survive longer in the cytoplasm to make it to a ribosome. So we have to have more stable RNA, and we'll cover that in the next section. So it is not a process where transcription and translation happens in the same, basically the same step. They are separate processes. We have to finish transcription before we can ever start translation for eukaryotes because we're complicated. So location is different. Eukaryotes also require transcription factors. So this guy, this guy, this guy, this guy, this guy, all of these are transcription factors, and we need those before RNA pol will bind. So eukaryotes, again, are complicated. We need transcription factors that will be in place before it's perfect and just right for RNA pol to bind. And then there are different types of RNA pol, and they're in different places. One of these is coming from the nucleolus, and the other 2 are found in the nucleus, and so it just, there are 3 different ones. I'm not going to ask you what the 3 different ones are and where they come from, but know that we are different from prokaryotes because our DNA is located in a different place, which means transcription and translation are in separate locations in the cell. We have to have transcription factors versus prokaryotes that don't need those. And prokaryotes only have 1 type of RNA pol, and we have 3. So that finishes this video. I'm going to go into the next 2 sections in the next video.